Good evening, all. Welcome to the Linnean Society of New York's first meeting of 2026. I'm Peter Davenport, acting president of the Society, and I hereby call the meeting to order. We're looking forward to a fantastic talk tonight with Anya Auerbach, titled Understanding Evolution Through Madagascar's Spectacular Radiations of Birds. More on that in a few minutes. We're here in person at the Liederkranz on East 87th Street. I got it right this month. But in February, we'll be heading over to the American Museum of Natural History for our first meeting there in a long time. So we hope we'll see you there in person. But to get into the museum then, you must register in advance. And please look for the instructions on our website. In March, we'll come back to the Liederkranz for our 148th annual meeting, which will feature the Eisenman Medal lecturer, Dr. Trevor Price, speaking on a bird's life in the Anthropocene. In addition to Dr. Price's talk, of course, the evening includes, for those of you who've been here in the past, a raffle with amazing gift bags, a silent auction, and of course, supper, and just meeting and socializing and getting to know each other better. We have great prizes lined up, and we are still welcoming donations. Consider giving tickets if you have them to the opera, sports, what have you, a few days at a vacation home, and handcrafts, interesting books. Please reach out to me via email or at president@Linnean.New.York to inquire about donations, and look for your invitation at the end of this month. We are a member-driven society, and you are the body and soul of our group. So please join me in welcoming 5 new members to the Linnean Society of New York. You saw a few others. We sort of got ahead of ourselves, but that's all right. So please, if you're here tonight, if you'd stand up just briefly to be acknowledged. We have David Campbell, Nicholas Caledon, Raymond Krause, Channing Wister-Jones, and Patricia Wong. Are any of you here? Thank you. Welcome. Thank you. Our members do everything voluntarily, and there are so many of you I should thank, but we'll leave that for a later date, at least for a lot of you. There are a few thank yous though for right now. Thank you to all of you who had given to the Birdseed Fund. We've raised nearly at least $800 since Sunday to fill the Central Park feeders, but we would love to reach 2,000 to avoid a fall plea. So if you haven't given and you're able to give a little bit, it's pretty easy to do, and the link is in the letter that I sent out on Sunday. Thank you also to the nominations committee, Dominic Ricci, Junko Suzuki, Suzanne Swicky. They've been looking for members to fill upcoming board vacancies. If you have any ideas, please reach out to them. Also, thank you to the board. We're looking to sort of restructure some committees. And I want to especially thank Kristen Ellington for stepping in for the Communications and Content Committee, focused sort of on online media. Miriam Rakowski, leading our membership and events committee, and Alan Drogan for agreeing to lead our technology committee. If you think you have skills or anything to offer, again, please reach out via email. I'm almost done, and then we'll get to the speaker. So we have, of course, trips coming up, including a trip to the Bush Terminal Park for gulls and ducks. That's still open, I think, until tomorrow? Yeah. Yes. Okay, so if you haven't registered and you're interested, join us. It's Birds and Brews. Starts at 10, I believe, and we will be birding until about 1, and then go enjoy some conviviality. Thanks to the Trips Committee for this and other wonderful trips. And finally, don't forget the Birdathon. It's high winter right now. Snow has melted, but the Birdathon will be on May 2nd and 3rd to benefit Great Gull Island. So start forming your teams now. But now I want to turn the meeting over to our Vice President for Administration, Karen Becker, who will introduce tonight's speaker. Karen?
All right. I haven't got this memorized, so I'm going to have to really look at my notes here. Oh, yeah, of course. Anya Auerbach is in the final year of her PhD at the University of Minnesota and the Bell Museum of Natural History in the lab of Dr. Sushma Reddy. Anya grew up in New York City, where as a nature-loving kid, she first got hooked on birding in Central Park. You may recognize her from Birders, the Central Park Effect, Jeff Kimball's movie. She graduated from the University of Chicago in 2019, where she wrote her undergraduate thesis with Dr. Trevor Price on the biogeography of birds in the Northwest Himalayas. Natural history museums have played a key part in her academic journey, particularly AMNH. From 2 summers with Great Gull Island in high school, her first research experience in the ornithology department in college, volunteering as an explainer in the biodiversity halls, and now as a visiting researcher using the collections as part of her PhD. In addition to research, Anya is a committed teacher and an organizer with her graduate labor union. She still identifies as a New Yorker and misses its glorious diversity of people, food, and birds, especially seabirds. Anya.
Awesome. Thanks so much for the introduction. Thanks for inviting me to be here. It's really fun to talk in New York and to the Linnean Society. And I do not know how to make that stop, unfortunately. I guess I'll hope it doesn't continue too frequently. So yeah, as was mentioned, hi, I'm Anya Auerbach. I'm a PhD candidate at the University of Minnesota, and I'm going to be talking to you about incredible biodiversity of Madagascar, especially these adaptive radiations of birds. A little bit more in front of the camera. Cool. Okay, so I've already been introduced, so I'll kind of speed through this, but yeah, I grew up in New York City, birding here and on Long Island, where a lot of my family lives. Got my start in Central Park. I lived in Chicago for 4 years, so more amazing urban birding there. And then since 2020, have been living in Minnesota. Just at the end of each slide, right? You're gonna get a dramatic sum. My research is on evolution of birds in Madagascar. As mentioned, I work a lot in natural history museums with these collections. I'm affiliated with the Bell Museum of Natural History in Minnesota. And this is just some photos of some of the work I do. That's my advisor, Sushma Reddy, and myself on the right. And some of my lab group preparing specimens outside the Bell Museum on the left. So, a little bit of an outline of what I'll be talking about today. Switching it. There we go. So I'm going to talk a little bit about more generally Madagascar biodiversity, why it's so cool, where it comes from. Then I'm going to get a little bit more technical for a bit. I'm going to talk about the concept of adaptive radiation, which is really big idea in evolutionary biology. It's kind of central to the work I do. I'm going to talk about some of the research I've done for my PhD. And I'm going to talk about some new work, things that I'm doing right now, and sort of stuff that's going on in my lab, which is really just an excuse to show you a bunch of cool fieldwork photos from Madagascar. So we'll finish out with that. All right, so before I get into my own research, I want to kind of contextualize Madagascar. So Madagascar is often called the 8th continent. It is one of the most incredibly biodiverse places on Earth. It's in the Indian Ocean off the coast of East Africa. It's the world's 4th largest island, so it's about the size of Texas and has a population also a little bit smaller than that of Texas. And, you know, in addition to the biodiversity, I think we need to acknowledge, that this biodiversity is rapidly disappearing. Madagascar is one of the poorest countries on Earth. You know, both the subsistence economy, you know, a lot of the habitat destruction going on there, is because people are trying to survive. So Madagascar's biodiversity is incredibly threatened for subsistence agriculture as well as for extractive, you know, farming and mining, etc. And this has been made worse by widespread drought due to climate change. That said, there's amazing people in Madagascar who are working to understand and protect what's left, as well as to improve people's lives. So this is not, you know, something that's my area of expertise or that I'm going to go into a lot today, but it's something that's really important, I think, to acknowledge. All right, Madagascar is one of the most significant global biodiversity hotspots, especially when we think about endemic species. The vast majority of organisms that are found in Madagascar, whether that's birds or mammals or reptiles or amphibians or plants or insects, are found nowhere else on Earth and are often really, really unique and unlike anything else on Earth. So you know, over 100 species of lemurs, all of which are found only in Madagascar. Top right is the incredible satanic leaf-tailed gecko, which I was fortunate enough to see. Madagascar has sadly lost some of its largest and strangest animals. It used to be even weirder. So before human arrival, Madagascar was home to elephant birds, the heaviest birds ever, and the bird that laid the largest known egg, as well as an incredible array of giant lemurs, including terrestrial ones the size of gorillas, as well as these massive sloth lemurs that sort of hung from the branches. So how did Madagascar end up with this huge and weird array of unique organisms? To understand a little bit of how we got here, we're going to go way, way back all the way to the Cretaceous, over 100 million years ago. So through the Cretaceous, the continent of Gondwana, which was this supercontinent that had united all of the southern continents we have today, South America, Africa, Australia, Antarctica, as well as India and Madagascar, this continent was gradually breaking apart. So you can see that sort of happening over millions of years here. And so Madagascar and India were actually the last connected to one another until somewhere around 80 or 90 million years ago, when they'd been drifting north and then separated. And India kept on going north until it eventually crashed into Asia, forming the Himalayas. And Madagascar has stayed pretty much where it is ever since that time. And so Madagascar has been totally isolated for around 90 million years. And it also hasn't really moved around. It's been pretty stable. So back in the Cretaceous, Madagascar was home to some pretty incredible organisms too. We have awesome fossils of dinosaurs from the Majanga Basin in northwestern Madagascar, as well as actually some early birds. There's a giant frog called Beelzebufo. But you know, these and many of these are most closely related from that time to the animals that we find from the Cretaceous in India. But you know, those guys died out along with the rest 66 million years ago. How did we get to Madagascar's living biodiversity? And unfortunately, fossils have been really unhelpful here. We have these wonderful Cretaceous fossils from Madagascar, and then we have no other fossils until recent subfossils that are just a few thousand years old. Which have been really cool for telling us that there used to be elephant birds and giant lemurs. They've not been so helpful for helping us understand how they got there. So a few of the organisms in Madagascar seem to have actually been there since the breakup of Gondwana. These turtles and crayfish have their most recent relatives in Africa and South America. It turns out the frogs actually are probably more recent and are maybe have not been there since the Cretaceous. But for the vast majority of Madagascar's living biodiversity, it's had to somehow manage to disperse there, right? Tell me if I should stop, but I'm going to keep going unless they tell me not to. So somehow or other, all of these things have dispersed over the ocean. Dispersing over the ocean's hard, right? We know it does happen. We could have seeds or tiny larvae on the legs of birds. A lot of these herps and mammals probably had to get to Madagascar by rafting on vegetation from the mainland, mostly from Africa, since it's a lot closer. And of course, that's really unlikely, right? But you give things 60 million years and unlikely things will happen sometimes. So For example, mammals have only managed to disperse across the ocean to Madagascar 4 times, not counting bats, which can fly, so they have a bit of an advantage. So we've got 4 endemic groups of mammals in Madagascar: Hunter-X lemurs, Eupleridae, which are the Malagasy carnivores like the fossa, and these nesomoid rodents. And those are the only times mammals have managed to reach Madagascar. So, okay, we're here because we love birds. Let's talk about the birds. Madagascar is the only island other than New Zealand that has an endemic order of birds. So they're at highest taxonomic level below all of birds. And in fact, it has 2. So both of these are thought to have arrived, tens of millions of years ago. We've got the Mesites, which are 3 species. We think they're most closely related to pigeons and sand grouse and some of their relatives, as best we can tell. They kind of look like a pigeon, partridge, something. Those are the 3 birds on the left. And we have the cuckoo roller, which is just the 1 species, distantly related to things like rollers or kingfishers. That's really just unlike anything else. One of the bizarrest birds you could possibly imagine. Madagascar is also home to an additional 3 endemic families. We have ground rollers, 5 species, ascites, which are very weird passerines that diverge very early within the passerine order. And around 11, it's probably more like 13 species of tetrakas or Malagasy warblers. I have to say the ground rollers might, even though they're not some of my main study species, favorite birds in Madagascar, they're just so unique and beautiful and cool. And then, of course, we've got the rest of Madagascar's birds whose ancestors arrived more recently. They haven't had time to diversify as much, but these are just a few examples of them. So one interesting aspect of Madagascar's avifauna that big global birders might be aware of is that although Madagascar is really high endemism, there's over 100 endemic species in Madagascar, it doesn't actually have a ton of species. There's only a bit over 200 breeding species in Madagascar. And what this little plot here is showing is basically the number of species found on an island compared to the island size. And you can see that most islands follow this pretty predictable relationship. Bigger island, more species. Madagascar falls way below that line. It has fewer species than we'd expect based on its size. And that's despite having lots of habitat diversity. Madagascar has rainforest in the east, including the central mountain range, so it has a lot of elevational variation. And then it has this dry forest in the west that turns into this very dry, spiny subdesert in the southwest. And so we're not really sure why there aren't more species in Madagascar. A lot of the oldest groups of birds there have very few species, like only 3 Mesites, 1 cuckoo roller. Maybe past diversity was really different from what it is today. We just don't know. But this is a really interesting open question. My personal wild speculation is that this might be competition with some of the other clades, like frogs and a lot of the reptiles are way more species rich in Madagascar than they are elsewhere. But I have no actual concrete evidence to support that. Okay, so why study Madagascar's birds specifically? A bunch of possible reasons, of course, right? 1, we don't know a whole lot about them. Madagascar is not the easiest place to work. Just as one example, these are 2 species of birds in Madagascar that were totally new to science, just discovered in the 1990s. The red-shouldered vanga and the cryptic warbler, which has one of my favorite scientific names, Cryptosilicola andrianosoloi. It's very fun to say, and I've gotten very good at it. Cryptosilicola andrianosoloi. And of course, there's lots of other things we don't know very much about Madagascar's birds in terms of their ecology and their behavior and all of these different things. We also want to know more about them because we can to better protect them. As I mentioned, a lot of Madagascar's birds are really, endangered, especially a lot of those rainforest birds and a lot of the water birds in particular. Down on the left, that's guy who's the head of the Peregrine Fund Madagascar. They just rediscovered the dusky tetraka after about 25 years of it going unsighted. It's like 2 or 3 years ago. And then the reason that I'm really going to be focusing on today is they have this really unique, cool evolutionary history that I've kind of already been alluding to. And so that's what I'm going to be focused on for today. So why do I study Madagascar's birds? I'm an evolutionary biologist, and so my interests are in questions like how and why did this huge diversity of species that we see originate? Why do they occur in the places that they do and not elsewhere? These are macroevolutionary questions, sort of the patterns that we see in biodiversity that have emerged over many millions of years. So one question I'm really interested in, why is diversity so unevenly distributed? This is a fun way to illustrate that. This is an evolutionary tree of all birds. It's not the easiest one to read, unfortunately. But over here, the little dark area on the right, those are our paleognaths. So those are all the big giant flightless guys like ostriches and cassowaries, as well as kiwis and tinamous. They are the first split in the tree of living birds. So we've got our paleognaths, and then we've got all of the other birds. And despite that, despite all those millions of years that there have been paleognaths, there's only like 50 species of them, at least that are alive today. And then on the other hand, we've got our passeriformes. That's one order of birds, and that's more than half of all birds, right? Over 6,000 species. Why are there so many more passeriformes than there are paleognaths? So one particular mechanism, thinking about how do species arrive? How do we end up with these unbalanced patterns of biodiversity? Is this idea of adaptive radiation. So I warned you, I'm going to spend a bunch of time talking about this. I will do my best to make this make sense. So you might be familiar with some classic examples of adaptive radiation. These guys show up in biology textbooks a lot, and we're also birders. Probably familiar with like Galapagos finches or Darwin's finches, and how they have these different beaks that have evolved in the Galapagos Islands. The Hawaiian honeycreepers are probably my favorite example. They're just so mega diverse and amazing. And of course, this happens not just in birds. So the cichlid fish in Africa's Rift Lakes are another really classic example of adaptive radiation. Biologists love to study adaptive radiations. They give us this kind of discrete, extreme case study of biodiversity, and that can help us figure out, kind of untangle how we end up with so many species and such a diversity of species. So what exactly does adaptive radiation mean? Just to give a sort of definition, what we're talking about is in order to consider a group of birds in adaptive radiation, they kind of have to meet two criteria. They need to have evolved an exceptional amount of diversity in response to some ecological opportunity. So I'm going to try and break that down. I'm going to talk about Hawaiian honeycreepers because you probably are at least a little familiar with them, and they're super cool. So Hawaiian honeycreepers, exceptional diversity. Maybe kind of obvious for one thing, at least before human arrival, there were over 50 species of Hawaiian honeycreepers in Hawaii. We're left with less than 20 now. But it's not just having a lot of species, right? Here's another example of an impressive radiation. Turns out that the different populations of what was formerly considered the rufous antpitta have been isolated from one another in different parts of the Andes Mountains for a really long time. In 2020, this study showed that they were different enough from one another that they should really be considered 12 different species. But they basically all just look like this, right? And they forage pretty much for the same food in the same way in similar habitats. You know, they're not exactly the same as one another, but they're really similar. Whereas the honeycreepers also have this amazing diversity of beak shapes that correspond to very different diets, very different foraging behaviors, right? Some of them are crushing seeds, some are probing for insects, some of them are sipping nectar. And that's why that second part is so important. When we say adaptive radiation, we mean we've gotten so many species because they're adapting to these different ecological niches. We're not just getting more species, they're doing different things. And so that's where that ecological opportunity comes in. This could mean a lot of different things, but in the case of Hawaiian honeycreepers and a lot of these other kind of classic examples, adaptive radiation was kickstarted because their ancestors arrived on this isolated island that didn't have a lot of competition, right? So the ancestors of Hawaiian honeycreepers, their closest relatives are rose finches, which are kind of like our house finches, that most of the rose finches live in East Asia. And so something that looks more or less like a rose finch arrived in the Hawaiian Islands somewhere around 6-7 million years ago. And there weren't a lot of other birds around, right? They got to Hawaiian Islands, there's like maybe nothing except seabirds there at that point. Maybe there's like Mohoidae, which are another extinct group. And And that gave their ancestors this opportunity to diversify. The finches on the mainland, there's all these other birds that already do all these other things, right? There's sunbirds that drink nectar. There's, all sorts of other birds that are already eating insects. And so finches have lots of species. There's hundreds of species of finches, and they're all trying to do finchy things, right? They eat seeds. They have short, stout bills for doing that. But when a finch got to the Hawaiian Islands and no one else is there, that's an ecological opportunity. They diversified to fill all of these different niches. But one thing that makes this interesting is it doesn't happen every time. So here we've got an elepaio. These are monarch flycatchers. They're another group that's made it to Hawaii. But elapaios, there's one species on each of the main islands, and they all look like this. And that's all of the elapaios have done, even though they've also been on the Hawaiian Islands for a good amount of time. So one thing we want to understand is, what is it about the specifics of a time and a place, but also maybe the specifics of a particular species, that ancestor that arrived there, that means sometimes you get an adaptive radiation and sometimes you don't. Why does that happen? Okay, so I've been talking about these really famous examples, but I work in Madagascar. I don't study those guys. Unless you're a big global birder, you might not have heard of Madagascar's 3 biggest radiations of birds. So I'm going to introduce them to you. Vangas, tetrakas, and couas, as well as their closest relatives that live outside of Madagascar. So first up, vangas. Vangas are a really spectacular example of adaptive radiation, rivaling the Hawaiian honeycreepers in my and many other smart people's opinions. The 22-ish Malagasy species are so different that before molecular phylogenies, before we started sequencing DNA to study how things are related to one another, they were classified as all different kinds of birds that were not related to one another because they just look so different. So to study how these guys have evolved and what makes them so unique, I kind of want to compare them to something else. And so I'm also studying their closest relatives that don't live in Madagascar, that we don't really think are an example of adaptive radiation. So in this case, that's what non-Malagasy vangas, the rest of this family, which are a whole bunch of birds that kind of look like shrikes. They are related to shrikes, although they aren't shrikes, that live in tropical Africa and Asia. So unfortunately, these guys might not be super familiar to a lot of birders who haven't visited that part of the world. A lot of them have shrike in their name. We've got wood shrikes and helmet shrikes and flycatcher shrikes and shrike flycatchers, which are different. Next up, we've got Bernieridae. I mentioned these guys earlier as tetrakas. These are the Madagascar warblers. Again, they're diverse enough that they used to be put in a few different families before we made a phylogeny a number of years ago. Some of them look very warblery and flit around up in the canopy. Others of them are chunkier and hang out in the understory. One of them even hops around on the ground. Their closest relatives, so their very closest relative, super weird. This single species family called the Donacobius that lives in South America. How did they get there? No idea. Super weird. And then their next closest relatives are this big family of warblers, the Locustellidae, that are found again in Africa and Asia. Last up, we've got couas. These are a big subfamily of cuckoos. They maybe aren't as diverse as the other groups, but still do a bunch of different cool things. Some are terrestrial, some are arboreal. The relatives are these Asian ground cuckoos, and then also the cuckoos. So what do I want to know? Talked about adaptive radiation generally. I've introduced the birds I'm studying. Very generally, what I'm interested in is, how has this exceptional diversity of radiations in Madagascar evolved? I'm interested in thinking about that in terms of the diversity of their morphology or their anatomy, their shape, because that relates really closely to what are they eating and how do they catch it. I'm also interested, although I'm not going to talk so much about this today, in their environmental niche. So what kinds of habitats are they living in? And then I'm also interested in thinking about, how are the radiations in Madagascar similar and different? Because we've got this really unusual situation where we have multiple radiations in the same place. And that really doesn't happen very often. So how are they similar or different to those mainland relatives I mentioned? And also, how are they similar or different to each other? And not just in terms of well, are they the same shape or a different shape, but in terms of the evolutionary history. How did they get there? Okay, so I'm going to talk a whole bunch about shape diversity in vangas because they are just so incredibly strange and diverse, right? As I said, these guys look nothing alike. We used to think that they were totally unrelated to one another, which is a very reasonable thing to think looking at them, right? We've got species like the sickle-billed vanga and the helmet vanga, and then some more subtle weirdos like the Lafresnaye's vanga in the top right there, which has a slight gap between its upper and lower bill. And it's a little harder to see here too, but they've also got diversity that's really interesting in other features. So I also looked at their feet because we've got a terrestrial vanga up top, the crossleaf vanga. And we've got the nuthatch vanga, which is called that because it behaves just like a nuthatch. And you can kind of see in that photo, it's got a giant hind toe, just like a nuthatch or a creeper does, because that's great for going up and down tree trunks. Look at that. So just wanted to really briefly try and like compare these guys to like other birds, because when we look for what sort of has an equivalent ecology and behavior to these things, we're looking at birds again in all different families. So here I tried to find some like North and South American equivalents. So we've got scythebills, which are oven birds or found in South America. We've got helmet vangas are kind of like a puff bird. It's another South American bird. Red-shouldered vangas are kind of like vireos. We also have some very warbler-like vangas. Crossleaf vanga, honestly, not a lot of easy comparisons. The closest I could come up with in North America was maybe an oven bird, but they're really more like these terrestrial ant birds in the Amazonia. And then, of course, nuthatch vanga is a nuthatch. So how do I actually study this diversity? Some really old specimens and some really cool modern technology. So my research is really dependent on the specimens of birds housed in natural history collections. Some of these specimens are really old. A lot of my specimens from AMNH here in New York are close to 100 years old. A lot of the Field Museum specimens are somewhat more recent, 80s and 90s mostly. And I think one thing that's so amazing is that when these specimens were prepared, people had no idea that in the future, I was going to be able to study the insides of these specimens using micro-CT scanning technology, or that other people would be able to extract DNA from even specimens that are 100 years old. So one of the incredible things about natural history collections, they're really like these archives, libraries of biodiversity, and they preserve this record of living things that scientists can keep going back to in the future with new technologies that, you know, the original collectors never would have imagined. So to CT scan specimens, I put them in little burritos to protect them and position them correctly. And then I put them in a CT scanner, which looks something like this. And what that allows me to do is I get this 3D digital image of the insides of these specimens, of their bones. And that allows me to study them in incredible detail without actually damaging these specimens at all. I sent all of my specimens back to AMNH, Paul. I believe undamaged. The downside is that processing these is actually really time consuming. So it takes a lot to go from not just a scan to actual data. And basically what I am doing here, once I have this 3D digital image, is I am placing all these dots on them. So they're called landmarks. I put all these dots in the same positions on each of these skulls from each of my specimens. I also did linear measurements of each foot bone, and actually pretty much all of the foot data collection and analyses were done by Ewan Lim, who's an undergrad co-author on this paper. And then basically it's a whole bunch of fancy statistics to turn this 3D, coordinate system, essentially describing the shape of the bird's bill and skull into data that I analyze. So this is what I sort of end up with. I have these three regions of the upper bill and the lower bill and the cranium, as well as the feet. So I'm not going to try and go into all the statistics, but I'll try to show you some of the results that I get out of this. Start with looking at shape diversity. This is an analysis called the principal components analysis, which I won't be explaining, but essentially what I'm trying to do is summarize. I've had like 200 something landmarks that are describing in a ton of detail what is the 3D shape of the beak and the skull of these birds, because they have all these fancy curves and hooks on them and stuff, and I want to capture all of that variation. But then I also need to kind of summarize it down. So these are just two axes that are sort of summarizing all of the variation in shape in these birds. And essentially what focusing on here is the triangles are the Madagascar vangas and the circles are the non-Malagasy vangas. And I'll just tell you that little sort of dash line kind of in the middle, that is showing you where all of the non-Malagasy vangas are. So all of the variation in shape and all of the vangas that are found outside Madagascar is captured in that one little area. And then some of the Malagasy vangas have kind of that same shape. They haven't really done anything different. But you can see a lot of them have gone off in all different directions and evolved really, really unique shapes. And a lot of that sort of most extreme difference is those ones that I pointed out before. We've got our sickle-billed vanga and our helmet vanga. But there's a lot of other Madagascar vangas that are also doing wacky things that are very different from the ones that are found on the mainland. We also see something a little bit different going on with the different parts of the anatomy that I looked at. So here I'm breaking it up. I'm showing you with the upper bill and the lower bill and the feet separately and variation in the shape in each of those. And basically, we've got some similarities. Obviously, that giant sickle bill, it's really weird in both the upper and the lower bill, but other things are different. The feet, no surprises. The two weird feet guys, the terrestrial one and the nuthatch one, they're like super different from all of the others, right? They're really far away from all of the others. And so I think that, to me, kind of the takeaways here are, Malagasy vangas are way more diverse. But we can also start digging into a little bit of how are they more diverse? What does that actually look like? And different species are unique in different parts of their morphology, right? The nuthatch vanga has a pretty normal bill, but those really weird feet. And a helmet vanga has a super weird upper bill, but its feet and even its lower bill are pretty normal. Okay. So I wanted another way to like dig into sort of what is going on here. How did all this diversity originate? Is to look at it on an evolutionary tree. So this is a phylogenetic tree, an evolutionary tree of all those vangas. I put their little skulls next to them rather than the birds themselves. Essentially, just to orient you, what we're looking at here is in the top half, we've got all of our non-Malagasy vangas in purple, okay? And then the bottom half, we have all of our Malagasy vangas, and they're in some different colors. And so, thank you. So here's our origin of the Malagasy vangas. Here's when they arrived in Madagascar. The other thing I'm showing you at the bottom is basically like, when is diversity originating in terms of their bill shapes? And we can see from sort of where is that black line relative to the gray, they're not really like generating a whole lot of diversity actually when they first arrive in Madagascar. Just kind of surprising, because like they got to Madagascar, there's not a lot of competition. Why isn't like tons of diversity originating right away? Then what happens is we have this origin of different ways of foraging. So here's what I think is going on here. Your ancestral vangas, almost all of the ones outside Madagascar, and a lot of the ones that sort of split off early in the group in Madagascar are gleaning. So gleaning birds are ones that kind of move through the undergrowth of their canopy, and they're picking food, in this case mostly insects, off of leaves or branches. And so when the ancestral vanga first arrived in Madagascar, it started to diversify in terms of different ways of gleaning. So for example, we have gleaning on the ground instead of on the branches, right? But then at some point, they evolved a new way of foraging. We see the origin of this sallying group. And that opened up a whole bunch of different new possibilities. There's a whole bunch of new ways to sally. There's a whole bunch of new ways to be a vanga once this sort of realm of behavior is available. And then at some point from that group, we have yet another way of foraging. We have probing. So sallying, that's kind of like you like fly out to pick food off the surface or maybe out of the air, like a flycatcher. Those are salliers. Probing is when you're like really reaching into like crevices in the bark or in leaves. And so this is telling us that the origin of these new behaviors opened up these whole new ecological opportunities over again, and really have like multiple rounds almost of radiation when these new opportunities open up for them. Okay, so we're talking a whole lot about like really macro stuff, maybe feels a little bit hard to follow. It's, this is like stuff happening over tens of millions of years. I'm going to zoom in now and talk a little bit more about like speciation itself, which might feel a little bit more familiar to a lot of birders because we think about like, ooh, are we going to get like new species splits and stuff, right? So as birders who, are looking like, oh God, we have eastern and western warbling vireos now or whatever, you're probably familiar with this idea of cryptic diversity, species that can look almost identical, but are genetically, maybe, ecologically different from one another. And some recent work from my advisor and some of her former students has shown, for example, that this rufous vanga that's found all over Madagascar, well, actually the ones in the different biomes in the eastern and the west are pretty different from one another genetically, and they should probably be split into like a dry forest and a rainforest species. She also looked at these Newtonias, these little warblery vangas, and found that the common Newtonia that's all over Madagascar is actually like really similar all over Madagascar, but the dark Newtonia that's found only in the mountain rainforest is split by this gap in the mountains in the north and south. And so I'm doing something pretty similar to that, slightly updated methods, focusing on another of these species from these radiations, the crested couas. So this is one of these arboreal couas. It's a cuckoo that bounds around up in the trees. It's found all across Madagascar's major biomes, and it has described subspecies from these different biomes. So we thought it was a really good candidate to look at more closely. And in particular, actually, the ones in the southwest have these rufous undertail coverts, the subspecies Pyropyga, and they've sometimes been considered a separate species because they are a little bit more different in terms of their appearance. Okay, so I want to think about how are these populations related to one another, right? And I'm going to go through this really quickly, but essentially, we use these like little bits of DNA from across the genome that are called UCEs. There's lots of different algorithms for building phylogenetic trees that matters which ones you choose. Here's one of the trees that I made. Essentially, what we're really the key thing to see here is one, yes, we have these groups. These subspecies turn out to like actually work out genetically for the most part. But actually, Pyropyga is more closely related to these other Western birds, and it's the Eastern ones that are the most different, genetically speaking, even though they look really similar. This is just to say that different methods can give you different results, and this is why science is hard. There's some other evidence that makes me think that tree is actually the right one and not the one on the left. Don't worry about it. There's another way to look at this genetic data is to cluster individuals based on genetic similarity. So when we cluster them, we again see these really discrete clusters, and that's another way that we can think, these seem pretty distinct from one another, right? These groups aren't overlapping. They're not like super close to one another. That seems like maybe it's evidence that they could be different species, or at least they're well-differentiated populations. One little sticking note, though, is that actually this one, which clusters closer to the yellow group, in the tree, it was part of the red group. So something a little funky going on there. And we can dig into that a little bit more if we map this on the tree. So here, I'm allowing individuals to be assigned split ancestry, basically. An individual could be a mix of two different populations. And so what we see is kind of what we'd expect, that at the boundaries between these biomes, we find individuals that have mixed ancestry from these subspecies. And so we see that, yeah, we actually are finding subspecies that line up with these biomes, that the ecology is structuring the genetics of these birds, and that there seems like there is some mixture, especially that one individual that I pointed out before is very mixed. And here's just another way of looking at that. And this does match up with what we kind of thought based on the phenotype. These are just pictures from eBird. And if I look at pictures in eBird from that kind of spot where we have that individual with mixed ancestry, they have like slightly reddish undertail coverts. Just, maybe what we would expect. So are these separate species? I'm not sure yet. I haven't decided. There's more stuff to do. I want to look at the timing of these splits, and I also want to do some more analyses that get more directly at, well, like, are there actually hybrids, essentially? So stay tuned. Okay, so what am I doing now? I mentioned that I want to compare patterns across the vangas, tetrakas, and couas. Unfortunately, I'm still working on my PhD and I don't have results from that yet, but I'm going to tell you about what I've been doing for it recently. And we've been doing fieldwork for future projects. Got to go to Madagascar in September, so I want to talk a little bit about that and, show you pretty pictures. Okay, so For the last month, I've spent a lot of time visiting museums to collect the final pieces of data for that study where I want to compare all of these radiations to each other. So this is a whole lot of going into natural history collections. They tend to look kind of like this. You're hunting all over the collection, trying to find the birds that you're interested in studying and going into these. So these rooms are climate controlled. You have these like really well-sealed cabinets to protect the specimens. This is at the Field Museum of Natural History in Chicago. Sometimes you have to climb up on rickety ladders to get drawers full of 100-year-old specimens down from high shelves. And then I bring them over to my little data collection setup. And I've got all these different things going on. So I'll tell you a little bit about it. I've got my collections guide at the top left. So that has a list of every genus in the collection and what cabinet it's in, because otherwise you would spend the rest of your life trying to find things. Sometimes things are classified under the genus name that they had 50 years ago, and then you have to go figure out what that is. But then I've got my calipers for taking linear measurements of the specimens. But I have a fancy newfangled technology set up with my calipers. So I have a foot pedal that enters the number from the calipers directly into the spreadsheet on my computer, so that I don't have to type them all in by hand, which saves me countless hours of work. However, I do have to type in by hand the measurements that I take of the wing and the tail. I do those with what's called a wing rule. And then on the right, I take photographs. So I take these standardized photographs next to a little ruler. And that is, I'm not doing 3D measurements like I did for the vangas because it's too time consuming for this many specimens, but I am doing 2D. That allows me to sort of describe the shape of the bill of those beaks and feet still better than I would otherwise. And it's just like, I have to spend a minute talking about what an incredible privilege it is to work with these specimens and these collections. You know, a lot of these things, as I mentioned, are really old, are, you know, these really priceless representatives of their species. I think I'll shout out especially the middle bottom are Cryptosilvicola randriana soloi. See, I got to say it again. And then some of the, well, only a handful of specimens that we have of that species there has at the Field Museum. These specimens were used to help describe that species formally to science. You know, we're really careful with collection these days. You know, it's an important document of biodiversity, and we also want to be really careful with it. So these are some of the only specimens of this species that we have. Most of the others are actually in Madagascar. So it was really special to work with those. And they were in fact collected by my collaborators who I got to meet when I went to Madagascar. So another project that my lab has been working on is wanting to study more about this biodiversity of these birds by studying their whole genomes. And for that, we want new samples that have really high quality DNA that will allow us to get those whole genomes. And for that, it's really awesome to have blood samples, actually. So we went on this sort of preliminary expedition to collect some samples. Back in September, I got to go to Madagascar for the first time because I started my PhD in 2020, which was not a great time for planning international field work. So I finally got to go this past September for the first time and meet incredible people and work in incredible places. So I know I'm short on time. I'm going to kind of like really quickly go through a whole lot of photos. Sorry. So here's a little bit of what we did. We were misnetting birds. Basically, it's this very fine mesh net you set up in the forest. If you're lucky, the birds don't see it and they fly into it and get tangled. And then you very carefully untangle them. And sometimes, like this guy, they really grab onto the net and you have to convince them to let go with their little teensy toes. And once we've extracted them from the net, we take some measurements. We also took some standard color photographs. So if people want to study like the plumage color in the future, they can do that. And then we took a little blood sample from the bird and we hold a little cotton swab to them until it's clotted, and then we let them go. And we mark them so we don't take multiple blood samples from the same birds because you do catch the same bird again sometimes. No, be free. This is our setup in the forest. It was incredible. I mean, we were out in the field for 12-hour days because we're near the equator, and so there's 12 hours of daylight. And we were out there the whole time, and it was just incredible being in this forest like that. It was so beautiful. And we caught a lot of really cool birds. So these are some of the vangas that we got. These are all my phone photos because I still haven't processed the photos from my actual camera. But yeah, this is red-tailed vanga, dark Newtonia, hook-billed vanga, blue vanga. We've got a crossleaf vanga, that's the terrestrial one, and Poland's vanga. And yes, blue vangas are that blue. I know it looks fake. It is real. They're that blue. It's crazy. We also got quite a few of those tetrakas, the Malagasy warblers, and one single couas that came into the net the very first day, and we never got another one, but it was so cool. That's my advisor, Sushma. Very happy with the couas. We also got members of some of those other endemic groups in Madagascar. So some of the ascides, we've got velvet ascidi and common sunbird ascidi. We got one ground roller, which was, again, kind of maybe my favorite. And then kind of stole the show. We got a cuckoo roller, which rarely fly into nets and get caught. So that was incredibly exciting. There's like one tissue sample of cuckoo roller in collections right now. So now there's two. We're very happy about that from a research standpoint. And of course, lots of other amazing endemic birds. And the forest was full of all these other incredible creatures. I could have done like 20 slides of insect photos. Madagascar is just, as I mentioned at the beginning, so incredibly biodiverse in all of these different ways. So, chameleons and other lizards and frogs, and we saw 11 species of lemurs. And these guys were just visiting us while we were doing field work. Both of these photos were taken like from where we were doing our work. And as I said, insects, really cool mammals. That's a red forest rat, which actually one of my friends studies. And a ring-tailed Vontsira, which is one of the Malagasy carnivorans. It's not a mongoose, even though it looks like one. And I'm at time, which is good, but I need to take a minute to talk about the people, because this kind of fieldwork is a really huge team effort. We try really hard to avoid engaging in what we call parachute science, where Western researchers come in to poor countries and do a bunch of science for themselves and don't really give back to the communities there. So we're doing everything we can, both to just financially support people who live near these protected areas so that hopefully there's an incentive to keep protecting them, and also to actually work with Malagasy scientists. So we have collaborators at the university there. There's grad students who are in the field with us. And of course, like literally we can't do anything without them. Like I don't speak Malagasy. I don't know where anything goes. Just like the team effort required for these things really can't be overstated, especially when we're like in the remote areas camping, and we had to go grocery shopping for 11 people for 5 days in the market, and then we have cooks with us and just so many incredible people who we got to meet. Got us shout out this guy, especially Narendra was our expert bird guide, and I did a bunch of point counts to sort of complement the mist netting stuff. And to be clear, Narendra did point counts and I wrote down what he told me. And I don't want to pretend that it was anything else, but he was also just such an incredible teacher. I asked him to quiz me on the birds and he did. He would be like, what's that singing? And I would like try and figure it out. And I got better, all thanks to him. So that was really awesome. And I talked about the fieldwork folks, but of course, all the rest of doing research, PhDs are hard. PhDs in this day and age are maybe especially hard, although I don't want to put down people who did them in the past. So, all of the scientific support and just people support. I think all the people at different institutions who go into this, like I ended up doing CT scanning at like 4 different institutions because our machine kept breaking. And then just friends and family. These photos are fun. That was my lab group in 2020, having grad student meetings on Zoom. And the Vanga stuff that I talked about is actually out. So if you are interested in reading the scientific paper, it's there. And with that, I'll take questions. Thank you so much for being here. And if your question is, can you show us those photos again? Because they're pretty. I will accept that as a question. Yeah, no one has studied color in any of these groups specifically. There has been a lot of really cool research on bird color that's been done recently. And one really cool thing about bird colors is they can be formed in different ways. So some of them are pigments, some of them are structural. All of those blues are structural color. And one thing that's really cool in the vangas is they have that in their feathers, which doesn't go away when they die. The feathers keep that color. But you might have seen with that blue vanga or the helmet vanga, they also have a blue beak and even a blue eye. And those colors don't stay when they die. The specimens of those species that have those bright blue bills have black bills. And so we think what's going on there is basically there's fluid in the bills that is changing the structure. And so when they dry out, when they die, the structure changes and it doesn't look blue anymore because it's these microstructures making it look blue. But we don't really know. And I would love someone to study that. And it's actually something my advisor is interested in, but hasn't had a chance to do. Is there anyone on Zoom? Yeah, the question was about there's been recent work showing really rapid change in bill shape in dark-eyed juncos, and this has been looked at in other species. I mean, really famously, actually, Darwin's finches were studied for rapid evolution in bill shape. And basically, what it seems like is, these changes that they're studying are real, but they're very small, right? They're not things where you'd like look at the junco and go, oh, whoa, its bill shape is so different now in a couple of years, right? So changes in like exactly how long versus how deep those sort of like linear changes in dimensions seem like they can happen pretty easily. And that's probably because, right, that's a relatively straightforward thing of like grow a little bit more this way versus a little bit less, right? I think it's really interesting to think about, especially in terms of something I think that study's getting at with how much of that is environmental, how much of it is plasticity, can change just due to getting more food or less food, versus how much of it's evolutionary encoded in their genes. But it does seem like the scale of change that we see with these birds, where it's like, now it's a long sickle bill. That's not so easy to do, right? Evolutionarily, you've got a lot more going on there than, well, this one's a little thicker, this one's a little thinner, which I will say, personal biases, obviously. It's one reason I think that the vangas and honeycreepers are so much cooler than the Darwin's finches. Because the Darwin's finches basically just have like thicker bill or thinner bill. Sorry. There's Zoom question.
A Zoom question. Are all the samples you studied from natural history museums, do they have living species hanging out in Madagascar?
For all the species I studied in natural history museums, also, sorry, I guess I don't need to repeat this one. You all heard it. So there's one recent extinction among these groups, which was the snail-eating couas. And at least within sort of scientific record, it was only found on this one island, actually, off the coast of Madagascar, the smaller island. And it was actually really unique looking, like totally different plumage pattern. It apparently specialized in eating land snails. And so it actually is in my data set. However, I did not collect that part of the data. Previous student did and then never did anything with it, which is why it's my data now. But I have not gotten to actually like hold the snail-eating couas specimen, and I would like to. Oh, yeah, I'll leave this up for a minute. I know there was a question in the back.
Follow up to the color question, and might not be good, there's not much of an answer to it, but I just wanted to bring attention, and you can see there's a picture on the screen, process of the ground going. I just noticed the facial pattern is very similar to a couple other birds that see pictures of from Southeast Asia, right? The red babbler and the barred rail. So I'm wondering if there's some sort of, I'm just putting out the idea of maybe there's some sort of advantage to having a stripe, having a white stripe in that particular part of the face for ground or in forest, tropical forest species?
You certainly see a lot of those really complex facial patterns in a lot of terrestrial birds, and it definitely looks bizarrely similar to, that ant bird that I showed earlier, to a lot of the like terrestrial babblers in Southeast Asia. The convergence is definitely super weird. I mean, it was classified as a babbler from sort of related to these Southeast Asian species before we realized it was a vanga. You do get a certain amount of convergence that's maybe just going to happen because birds have tracts of feathers, right? So the feathers aren't just like sprinkled evenly over the body, they're in these distinct groups. And so often like the genes encoding color will basically sort of operate by tract. And so you get a little bit more of like, oh, things that are not related look more similar than you'd expect just because of, it's not just totally random that way, right? But yeah, I also definitely think that similarities in plumage and pattern especially are something that haven't been studied a huge amount just because it's hard, right? Color is easier to quantify than pattern is once you start getting into like complex variation in patterns. Yeah, I hope that was helpful.
So I'm curious about, you studied the morphology. How important is morphology for this DNA sort of taking over? Because in the old days, humans were all studied exclusively like morphology, and there's
Yeah. Question was sort of about how important morphology versus DNA is, because of course morphology really dominated in the past and DNA has sort of come to the forefront now. Yeah, in the past, classification was based on morphology or anatomy because that's what we had right before we were sequencing DNA, or very early on, they were actually like doing protein electrophoresis. And nowadays, we don't pay very much attention to morphology for classification. We definitely do still take it into account if we're deciding, should these things be species versus subspecies, or, it's interesting to look at, like when they split ruby crowned kinglet into its own genus from the rest of the kinglets. One thing that came up is it has a different scaling pattern on its legs than the rest of the kinglets. And that difference in anatomy was something that people did consider. But I'd say that overall, we don't pay all that much attention to anatomy when we're trying to classify things anymore, because DNA is just so much more accurate for that. And that is because anatomy, morphology, phenotype are so important and interesting when it comes to actually understanding the ecology of the birds. What do they do? Like, how do they live? How are they, all of these other things. And so because those, things that live in similar places tend to look similar, that's maybe a reason to not use anatomy for classification, but it's a reason that it's really interesting to study for other reasons. And so I pay, a ton of my research is looking at this anatomy, because what I'm really interested in is how have they diversified in terms of their ecology, in terms of, the different ways that they're foraging, the different places they're living, all of those things. So I will say, overall, the field has moved very strongly in the direction of like genomics is God, which is correct in some ways and a little frustrating in other ways, especially when I'm looking for postdoc jobs right now and everyone just wants me to be able to do genomics, which I can sort of do, but probably not as well as other people who've done nothing but genomics for their PhDs. But I think it's so important to look at, what does the organism actually look like when it's alive still? Behind you.
Something with between Madagascar and New Zealand. When you go to town to buy food, anybody doing lemurs?
Any what?
Lemurs? Lemurs.
No, question was, anyone offer me any lemurs, because there is a market for their meat. No, The only live animals I saw in the markets were gray-headed lovebirds, parrot species in Madagascar, which are, I mean, declining but not particularly endangered. There is, people absolutely do eat a ton of the animals in Madagascar. It's a very complex issue, but it's mostly like local people eating it locally as like a source of protein because they're hungry. It's not something you see a ton being sold in markets, at least in like bigger towns. Yeah, I didn't see any. I believe that it's true in some places. One really interesting thing in Madagascar is there's a lot of local taboos around different foods. So in some places, eating lemur is basically taboo, and in other places it isn't. And this comes up with all sorts of different things. So when we were traveling to this more remote field site, there was a particular kind of lentil we were told not to purchase because that particular kind of lentil was taboo locally where we were going. Waiting for what I'm doing.
Yeah. Are there any genetic traits or behaviors that would indicate if a species is more likely to diversify and radiate more than another species in the same environment?
Yeah, so the question that I teased at the beginning, it didn't really answer because we don't have a good answer yet. I just think it's a really interesting question. Why do we get Hawaiian honeycreepers sometimes and Elapayos other times? So there's some hypotheses out there. One that's been put forward actually by Trevor Price for birds is that maybe granivores, so birds that mostly eat seeds, are more likely to radiate. Basically based on the fact that both Darwin's finches and Hawaiian honeycreepers have granivorous ancestors. Vangas don't. In fact, none of these birds do. All of them are primarily insectivores. It is interesting that they have not diversified in terms of their actual diet as much. So, right, we look at the Hawaiian honeycreepers. Some of them still eat seeds like their ancestors. Others eat insects. Others eat nectar. In the vangas, they're all insectivores except for the bigger ones that will also eat vertebrates, so like lizards or things. None of them have evolved to eat seeds or nectar or any of those things. But they've obviously still diversified a huge amount in how they forage for those insects, right? So that's one possible factor, hard to say. There's lots of other complex things about some of which I looked at in this study actually in terms of like how freely traits are to evolve independently of one another. So maybe if like you're only able to, if your upper and lower bill can only change together, maybe it's harder to diversify than if they can change independently of one another. But that's still sort of hotly debated within the field. Think back there's, yeah.
You mentioned that after radiation cichlids I was just wondering, do all cichlids have their own niche where they have their own food source
So yeah, not super an expert in cichlids, but I have actually read quite a lot of cichlid papers because I've read a lot of adaptive radiation papers. There's like hundreds of cichlid species in these lakes. It's pretty incredible. Questions about if like all of the cichlids have their own niches or not. So there's tons of different cichlid niches, but it's not that every single species has their own one. I'd say there's probably easily a dozen different niches, probably more. And some of them do really weird things, like they're specialized scale eaters of other fish, or specialize on eating just specifically snails. Others are vegetarians, or maybe they're filter feeders. So there's lots of different niches, right? And they have diversified into them really, really quickly. The cichlid radiations are only like a couple 100,000 years old, which is just mind-boggling how fast they've evolved. But yeah, it's not that every single species is unique.
Jacob asked, did you misnet the cuckoo roller at Iwohiboro? I went to Ranomafana in 2024, and then Evo Hiboro in 2025 for lemur research. But I'm also a birder and thought they were much more common in Evo Hiboro.
Yes, you are exactly correct. In fact, yeah, we were in Ranomafana National Park for most of the time, and then we went to Evo Hiboro protected area, which is this isolated forest fragment on the high plateau of Madagascar. So there's this sort of high, dry central plateau that we think used to be more forested, but a lot of that forest was lost in the last sort of 1,000 years of human presence. But there's these isolated fragments. And so we visited one of those, and indeed, cougar rollers were like really common there and a lot easier to see, maybe in part because the forest is a little more open. It is drier than the rainforest. And yeah, that's where we caught the cougar roller. We were like kind of chasing them around, trying to get them to go to the net, and had a couple of close calls before we got one. They're kind of too big for the nets we're using, so they don't get tangled in them very well. So it was basically a matter of calling it into the net and then immediately running up and grabbing it before it could get itself out again.
I was wondering, when you see birds who kind of deviate from their family and act like behaviors of other species, I thought this was because you kind of talked about when you were at the industry how names got changed over as and that happens a lot for a lot of birds. So I was just wondering if in your research and everything like that, or just if there's any like poetry and like why one bird in this family act like a
Yeah, so the question's about why we sometimes see these like particular species in a family just acting like something totally different from a different family. And in the case of the birds I study, at least, it comes back to that adaptive radiation idea and the ecological opportunity, I think. So, we look at the mainland and we've got Cetidae, we've got like this whole family of nuthatches that are like specialized to do nuthatch things. But no nuthatches or anything like a nuthatch, no woodpeckers either ever made it to Madagascar. And so there is this opportunity there potentially. And there's, of course, with evolution, an element of random chance too, right? The right mutations had to happen for, a really long hind toe to evolve in those birds, which, is increasingly likely when you give them many millions of years of evolutionary time for that to potentially happen. But it's that combination of, well, no other bird was doing that, so there wasn't competition to be doing that, and just chance of evolution. And it's certainly, ecologists and evolutionary biologists will get into heated arguments over whether the concept of an empty niche is real. The idea that a niche, a particular like way of living, as an organism exists sort of independently of an organism actually filling it. So can I say that there's an empty nuthatch niche that it filled in Madagascar, or is that just an absurd thing to say? Is something that ecologists and evolutionary biologists will get into fits over. But it's useful as terminology, in my opinion. Was there someone else on Zoom?
There's one more.
Okay. There are several more people on person, so we should probably go to an in-person.
When looking at adaptive radiation, are you chiefly looking at vertebrates, or thinking about the invertebrates, so that archaea or bacteria getting off with a much more rapid sort of rate of evolution? Is that relatively work, or is that sort of different?
Yeah, the question is about whether we're mostly thinking about vertebrates for adaptive radiation or like other kinds of organisms too. And definitely not just vertebrates. It does get complicated with bacteria and archaea because they're really fast, and they're also just so weird with like horizontal gene transfer and stuff. It's harder to sort of linearly trace the evolution of a clade. There's some really great studies on sort of like the early phase of what we'd consider an adaptive radiation, where you have just like 2 or 3 niches evolving in yeast. So there's, what are they called? Like smooth and wrinkly spreaders or something like that is like this classic research actually, that was done in yeast on adaptive radiation. And we certainly see it in like insects. There's like really cool radiations of insects in Hawaii, for example, and spiders too. Last person on Zoom. And then if you're in person, I think there's some talking time after this. So
You mentioned you are planning to do whole genome analyses. Will you specifically look at genes that code for bill morphology and not just UCEs?
The question was plans for the whole genomes, and I am not doing the whole genome stuff as part of my PhD, at least. There's a slight chance I could do whole genome stuff for Madagascar birds for like a future like postdoctoral research project or something. In which case, potentially. There's been really wonderful research looking at whole genomes of like Darwin's finches and identifying like the specific genes that are involved in differences in bill shape in them. However, it's easier in them because one, the Darwin finches are like really closely related to one another and they basically only differ in their bill shape. So it's a lot easier to look at where the differences in the genomes of the Darwin finches and go like, well, that must be for bill shape because they're basically the same in all of the other ways. Whereas these vangas are really different from one another in lots of ways. And so it's a lot harder to look at their genomes and figure out, well, the differences here are related to this thing, the differences here are related to that thing, because there's just so many differences across their genomes. And, you know, the genome does not come with neat little signs saying the bill shape's over here. So it'd be really cool to do that, but it'd be really hard. And it was actually something I thought about earlier in my PhD as a thing I would love to do, and decided that that would have to be like the entire PhD if I did it. And I was already doing this other stuff I was interested in. I think we're at time. I'm supposed to wrap things up now. I'm happy to keep taking questions, but
Thank you so much for your talk. We really appreciate it. And if people in the room have more questions, you know, mosey on up. Otherwise, Zoom people, it's to encourage you to come in person.
but thanks for your questions, Zoom people.
Thank you so much.